lots of different types of viruses that can infect animal cells. One of those types, retroviruses, has a particularly interesting life cycle that we will discuss now. Retroviruses are a unique subset of viruses that utilize positive sense single-stranded RNA. Positive sense refers to the ability of the virus's genome to be directly translated by the host cell's ribosomes. And these viruses only use RNA, not DNA, for their genome. Other viruses have RNA genomes too, but retroviruses have unique features that set them apart from other RNA viruses. Unlike other RNA viruses, the positive ssRNAs give retroviruses the ability to convert their RNA genome into double-stranded DNA, or dsDNA. Let's look at how that works. The process of converting ssRNA to dsDNA is called reverse transcription. Reverse transcription begins with the retrovirus entering the host cell like any other animal virus. And like any other animal virus, once inside the cell, the virus must be uncoated, which allows for the release of the ssRNA from the retrovirus. And from this point, things become a bit more interesting. Once in the cytosol, the ssRNA is transcribed by a virally encoded enzyme called reverse transcriptase, which uses the RNA as a template to create a new dsDNA. That's why this process is called reverse transcription. Transcription uses DNA to make RNA, but this process uses RNA to make DNA. It's transcription, but in reverse. From this point, the newly formed dsDNA is moved into the nucleus, where it will be integrated into the host DNA randomly. That is, it could insert at any location within the host DNA, with the help of another virally encoded enzyme called Integrase. This means that when the cell transcribes its own genes, it will also transcribe the virus's genes. The newly formed viruses can then exit the cell via budding, typically leaving the host cell intact. The retroviruses are also referred to as proviruses, since their viral DNA is integrated into the host's genome. Because of this, when the cell goes through the cell cycle, it will replicate the viral DNA along with its own DNA, and daughter cells will receive the viral DNA as well. This comes with an important consequence. You may recall that a transposable element, or transposon, is a gene that can move from one portion of DNA to another. Well, here we can see that this strand of DNA has a transposon, which means that this piece of DNA has the ability to copy itself and insert into a completely new area. Please note that some transposons can be cut out and inserted, rather than being copied and inserted. A different lesson discusses that process in more detail, but the important thing here is that when a typical transposon moves, it remains in the form of DNA the entire time. Retroviruses are believed to be the source of a different type of transposon, the Retrotransposon. Now, beginning with transcription, the viral DNA is transcribed and sent out to the cytosol. That means retrotransposons move around the genome via mRNA intermediates rather than staying DNA the entire time. Once in the cytosol, reverse transcriptase can bind to the viral RNA, allowing it to undergo reverse transcription. The newly created dsDNA can then undergo integration, where Integrase integrates the dsDNA into a new place within the host DNA. And that's retroviruses, RNA viruses that can alter your genome.